Thank you, Lila. Good morning, everyone. It is a pleasure to be here with all of you this morning, and a great pleasure for me to be the first speaker after finishing off with that wonderful session last night. So Lila set me up really nicely today. Normally, the first thing I do is stand up here and explain to you why I'm a biologist and why it's really important to care about biology. The bottom line here is that if you have a body, if you eat food, or if you live anywhere, you need to really be thinking about what's going on in the life sciences, because the things that are changing right now in biotechnology mean that the future of life is going to be very, very different from the past. And for me, this is super exciting, and I'm hoping after today it will be exciting for you too. So one thing I want to do today is work on this metaphor for something that I'm showing you here, which is this idea of a cul-de-sac. So I think yesterday we all saw that these exponential technologies that we're talking about bring with them very heavy ethical and moral problems. And I think, as we've seen a lot in politics lately, people are becoming very anxious about this, and they come into this cul-de-sac and it's a dead end, and the only way they can see to get out is by going backward. So I'm going to argue to you today that no matter what kinds of questions we raise, we don't get to go backwards. So keep this metaphor in mind as we're talking together today. So let's talk about biology. Just as a quick reminder from kindergarten science, all life on Earth is related. All life on Earth comes in the form of cells, including you. And if you zoom in far enough on the level of a cell, you get down to the level of the chromosome, which is kind of like the instruction manual for you or for any living thing. And if you zoom in even further onto the chromosome, you get down to the level of the famous double helix, which is the chemical molecule, the physical structure of DNA that's made up of 4 chemicals that for short we call A, C, T, and G. And it's the order of those chemical letters that contain the information that is the source code of life. So you have about 3 billion DNA letters in a very specific order in your DNA, in the human DNA. And 99.9% of your DNA is identical to the DNA of every other person on Earth. So that thought helps me get out of bed in the morning, so I'm going to say it one more time. Every human being is identical to every other human being on Earth, 99.9% of their DNA. Now, I'm a California hippie, so to me that means all of these reasons that we kill each other, we make them all up. They're not hard-coded in our DNA. So for me, that's a really exciting thought, because if we can cause problems, then we can solve them. So let's go back now to this idea of DNA as information. So when I think about a chemical molecule of DNA, which I do pretty often, confession, I don't actually see this chemical structure that I'm showing you here. I see this. I see computer code, because DNA is source code. And this is where the digital biology part of my talk comes from, because once we start thinking of DNA as information or as computer code, then everything we can do with computer code, we can also do with DNA code. So computers store information as zeros and ones. Life stores information as A, C, T, and G. So let's just think about that for a minute, right? Let's say I want to move big blocks of code around. Okay, well, in biology, that's called genetic engineering. We've known how to do that since the 1970s. Let's say I want to write DNA code from scratch. That's synthetic biology. We're going to talk about that a bit more today. And let's say I want to go in and I want to debug the DNA code one letter at a time. Well, I can do that using a suite of technologies called gene editing, the most famous of which is called CRISPR-Cas9. You've probably heard of it by now. And we're going to be talking a lot about CRISPR today, because it's a phenomenally empowering tool, but that brings with it some really interesting questions. So this wouldn't be a talk at Singularity if I didn't show you an exponential curve. So please note the log scale on the y-axis. So DNA sequencing technology is the process that we use to read out that order of A's, C's, T's, and G's in a genome. And you probably remember in 2001, the Human Genome Project and Singularity Genomics simultaneously published a draft sequence of the human genome. And back in 2001, so it was a draft, right? It wasn't even the whole thing, and parts of it were kind of crappy. And so that cost $2.7 billion to make and took 10 years. So 2.7 billion US dollars will get you a really fancy fighter plane, right? So from 2001, the price performance or the cost of DNA sequencing approximately followed Moore's law until 2008, when there was the invention of a new chemistry and a new type of computation in order to read DNA sequencing. And that technology was given the name next generation sequencing. We're actually on to the third version now. What do you guys think that's called? Yeah, I'm not responsible for that. And so after 2008, when next generation sequencing hit the scene, now you can see that the price performance of DNA sequencing began radically outperforming Moore's law until 2015, when it cost 1,000 US dollars to sequence an entire human genome. Now in the United States, that's cheaper than an X-ray, which says a lot about a lot of things in my country. But today, you can go and you can get just the active bits of your DNA sequence, that's called your exome, for about 200 US dollars, right? Some of you might have spent more than that on dinner last night. So this is a massive revolution in the way that this type of DNA information is becoming accessible to people. In 2001, when the human genome was decoded for the first time, it was done by these machines, I'm showing you some of them here, that were about the size of a refrigerator and cost at least $1 million a piece. And they still have these warehouses in Boston, these long, long buildings with no windows in them that were completely full of these machines. And these machines were what was used to sequence the human genome the first time out. This is what a DNA sequencer looks like today. One type of DNA sequencer. This is called a MinION. It's made by a company in England called Oxford Nanopore. And just last week, Oxford Nanopore also released a companion AI supercomputer that is also handheld. It runs on an NVIDIA chip, and that gives you real-time DNA analysis. So rather than doing your work in this giant warehouse, you've got a handheld DNA sequencer that plugs into your laptop and into a supercomputer that does the sequencing for you. There's also a new version of this sequencer going to be coming out soon called the SmidgeION, which plugs into your smartphone. So now ask yourself what this means. What does it mean in society in a day where everybody is walking around with a DNA sequencer in their pocket? This idea of privacy that we've been talking about later. Can you really protect your DNA? Well, some people say, okay, Tiffany, I'm gonna keep all my DNA private. And actually, to me, that doesn't make any sense, because you're leaving your DNA behind you on every single thing you're touching. You just drank out of a cup, your DNA is on that cup. You're touching your phone, your DNA is all over your phone. Stop touching your phone. I used to be a teacher, you can tell. So you're leaving your DNA molecules everywhere behind you, which means I could wait until the break, when you guys go out to get more coffee, and I could come in here and I could take the DNA off of everything you've touched, sequence it without your permission, and have your DNA code. Now, I'm not going to do that because I'm a very busy woman. But so for me, this idea of DNA privacy is actually a bit strange. And let's take this one step further. Who owns your DNA information? So how many of you have done like a 23andMe kit or something like that? Okay, who owns your 23andMe data? Thomas, who owns it? 23andMe owns it. 23andMe owns your data, which means you have no idea who they're selling that data onto, whether that data are anonymized, and how that data are being used by some other company. And so there's a whole new industry now called biobroking that's trying to flip that around and say, no, actually, I should be the person who owns my DNA sequence, and it should be up to me to sell that information on and to say what gets done with it. So I'm just going to give you a very brief example of this. It's a company called Nebula Genomics that's in Boston, and what they've done is they're flipping this model around. So in their model, they're saying, no, the individual is the owner of their own DNA, whether it's in their body or out of their body. And so they're setting up a marketplace where an individual can say, go to a pharmaceutical company and say, hey, pharmaceutical company, I know that you are working on this disease that runs in my family. Pay me to get my genome sequenced. And so then what you do is, using a blockchain-backed ledger, you are able to, as an individual, contact a sequencing facility, pay in some sort of crypto coin to get your DNA sequenced. The DNA information is then stored completely decentralized, so if there's a hack, nobody gets all the DNA. And then you get paid for that money for that sequence when you sell it on to a pharmaceutical company or to anybody else. And because all of this information is on a distributed ledger, you always know who's been asking for your DNA and whether they should have it. So this is a really interesting marketplace model that I'm following very carefully because it totally changes the paradigm of how we think about data, and in this particular case, DNA data. You know, we were talking about Facebook and Twitter and Google, right? What if Google had paid you for every one of those emails that you wrote over the last 15 years that let them develop those nice little suggestions for your email box, right? Totally different model. So let's talk about DNA as an information storage molecule for a minute, because by definition, that's what it is, right? So we've had life on Earth for, let's say, 4 billion years, we'll round, and all that time, DNA has been storing information. And so if you were to take all the data that we have now, and you probably know we have a data crisis right now, right? Too much data, not enough storage. So if you were to take the entire contents of the internet, all of it, even the pornography, and turn it from zeros and ones into A's, C's, T's, and G's, you could put the entire content of the internet in the back of a pickup truck. And if you kept it cold and dry, DNA stays stable for thousands and thousands of years, unlike your flash drive. So what this means is people are thinking more and more about how do we make DNA of any code that we want so that we can store information this way. The blue curve I'm showing you here on the screen is that same price-performance curve before. And the orange curves and the pink curves are data that were shared with me about the cost of making DNA from scratch. It's not quite 3D printing, but you can think of it that way for now. And so what you can see is the costs of making DNA is also dropping, but it's not dropping as fast as DNA reading is dropping. And so if you were to extrapolate out, you'd say, okay, well, it's going to take till about 2040 until it's as cheap to write DNA as it is to read DNA. But this is actually a false analogy. This is where Moore's law breaks down for DNA, because transistors can't make more transistors, right? But by definition, DNA makes more DNA. That's what it does. As soon as you boot it up into something alive, that living thing happily makes more copies. So you don't have to keep buying a new DNA strand every time you want to do this. So that means we don't have to wait until 2040 for the advances that you could do just by programming DNA from scratch. And I love being wrong in my job, by the way. Just a couple of months ago, a group published this method that is already off my curve in terms of how cheap it is to make DNA. And then on Friday of last week, a company announced that in San Francisco, that instead of just being able to make 50 letters in a row, which was the record 3 months ago, now they can make 150 letters in a row. So this stuff is happening faster and faster, cheaper and cheaper, and there's all kinds of really important implications for this. So for example, I talked about storing information. Well, it's not just like Wikipedia that you can store. So a group up in Harvard, actually the same group doing the Nebula Genomics, they figured out how to encode a movie, meaning time-resolved data, by taking it and transforming the movie, which here you see is the originals on the left. They translated that using the code I put at the top into zeros and ones, fed it to a bunch of bacteria, and then a few days later had the bacteria give them back the DNA. They decoded it, and then they got the image on the other side of the screen. It's not perfect, but you can see what it is, right? And so this idea of using DNA instead of silicon computer chips is really starting to take off, and I think you'll see a lot of progress here. So what I want you to do is I want to encourage you to think about life as if you were an engineer. Living systems, they take an input, they do something, that's the magic of biology, and then there's an output, and then you can reach a goal with it. This is thinking about biology as a synthetic biologist. And so my challenge to you is to think about your moonshot. The biology is the technology. Life has had 4 billion years on this planet to figure out how to do things way better than we know how to do them, right? So what you can do is if you think about living systems as input-output systems, then you can start programming life. So let me show you a really simple example that I use when I'm talking about this. So let's say you're me, and you're really excited about scuba diving, and you're really concerned about the plastic in the ocean, which, by the way, you all should be really concerned about that. So let's say I take a fish, and I genetically engineer that fish to be able to eat the plastic that's in the ocean. And then on top of that, I make it so that fish, when it poops out alkaline molecules into the ocean, thereby reversing the acidification of the ocean and saving the coral reefs. Thank you. Yeah. What could go wrong, right? So it's really trivial, and the devil's going to be in the details, but the biology is the technology. So I want you to keep thinking about this, and in fact, I want you to do more than think. I want you to go play. So there are biohacker spaces all over the world, which are like a gym that you buy a membership. But instead of having exercise stuff, there's biological materials in there. All of the DNA sequencers and all the stuff you need to do these techniques, they're available to you. You can go play. So that's my charge to you. Go play. Take your kids to go play. Let's talk about agriculture for a few minutes, because for me, one of the major things about the life sciences is how we're going to be dealing with the threats of an ever-increasing population and also climate change. And so this is a little awkward because I'm American, but I'm going to go ahead and say this. Climate change is real. Yeah, I know. It's an existential threat to humanity and to the rest of the planet, and it was caused by humans. Okay, there, I said it. So now that we can all agree about that, thank you, President Trump. So if humans cause this problem, to me that means humans can fix it, right? I honestly believe that. So agriculture is one of these places where gene editing in particular is poised to be completely revolutionary. Remember, we said gene editing was going in and debugging the DNA code one letter at a time. So let me show you two really simple examples of that. This cow lives up at UC Davis, which is up the road from San Francisco, where I am. And this cow was born without horns. So normally when you raise cows together, cows do all kinds of crazy things to each other, including fighting and poking each other with their horns. So the farmers always have to cut the horns out of the cow's head when it's still a baby. As you imagine, that process is unpleasant for everyone. But with using gene editing technology, this cow never had horns to begin with, so you don't have to dehorn it. And while we're thinking about cows, I want to set you up to listen to my colleague and friend Lowry later in this session, because imagine a day where we don't have cows at all. It could be coming. Now, here's another nice example of a group that was looking at tomato plants. And what they realized is if they took their DNA eraser with CRISPR and they just erased 12 letters of DNA, they could make a tomato plant flower earlier. And if you can do that, that means on a single piece of land, you can get more crop cycles out of a single piece of land without needing more space, without needing more crops. And if you get more tomato flowers, that means you get more tomatoes. So for me, this was really exciting because this is showing me that these tools are able to radically transform our ability to face climate change and expanding populations. And on top of that, it also had the ability to change the GMO debate. So let me be very clear. This is where everybody looks me in the eyes for just a second. There is no credible scientific evidence that GMOs negatively affect humans. It's just marketing, okay? People have been looking for 40 years. There's no evidence of it. And so when this CRISPR technique came out, people said, well, hey, great, CRISPR's not moving DNA between organisms, so all this GMO nonsense should go away, right? Because it's a totally different process, and I was really excited about that. And instead, you in Europe got this in July, the ruling of your Court of Justice of the EU claiming that CRISPR organisms are GMOs, which not only fundamentally misrepresents the science, it's like taking the magic tool that's going to save humanity and throwing it away. I was really disappointed by this. And in fact, I'm not the only one who's disappointed. If you're thinking about the future of innovation in the EU, you should be really concerned about this. Because folks who want to solve these problems and who want to do this research, they're going to leave and they're going to do their work elsewhere. And that's a big problem for the EU. Now we can talk a bit about whole species, right? Not just changing corn, but what about animals? This one I'm kind of excited about. So there's a group there's this thing called resurrection biology, where people are taking, not velociraptors to my knowledge, we all saw that movie, we know what happens, but they're taking animals that can't eat people and resurrecting them in new ways in order to both increase biodiversity, but also to have some interesting effects. And in fact, just this morning, an article came out in Science that was talking about how having large grazers, like woolly mammoth, could convert tundra back to grassland, which is not only a major carbon sink and is going to help with climate change, but also reduces the level of severe wildfires. And I know that you all had record-breaking wildfires this year. We had them as well in California. And so if we could use these types of things like woolly mammoth to return our tundra to protect the permafrost and to make climate change a little less severe, I think that's not only really cool, the idea of getting woolly mammoths back, but pretty useful too. Let's talk about mosquitoes. What if I told you that I could engineer every mosquito on the planet to never be able to give another human being malaria, or dengue, or Zika, or yellow fever, or Japanese encephalitis? We can actually do that through a technique called a gene drive, and they're actually already happening around the world. And the reason this is so important is if you do the math on this, and all things being equal, you could push a gene drive through every mosquito on the planet in about 11 to 14 mosquito generations, which is a little more than 1 year. Okay? In fact, you could even push a self-destruct mechanism through every mosquito on Earth in a little more than a year and cause all mosquitoes, well, all disease-causing mosquitoes on Earth to go extinct. So we're really good as a species. Humans are really, really good at wiping creatures out. But we're finally at the point where we can talk about doing it on purpose. And I think you can see that this raises a whole lot of questions. Now, no one's sitting around and just worrying about this. This is already taking place. A town in Australia has already used a slightly different kind of genetic technique to make it so that they had no cases of dengue in their town, which is spectacular. But if I just told you that we can edit plants, and I told you that we can edit mosquitoes, and I told you that we can edit fish, obviously that means we can edit humans too. It's the same molecules. There's no difference chemically between human DNA and mouse DNA or mosquito DNA. So if I could use gene editing to, let's say, get rid of the gene in a cancer that tells a cancer cell to grow, I think that's a pretty obvious win, right? If I were an adult and I had a cancer, I could rub a cream on it, the cancer goes away, I'm in good shape. I think we can all agree that that would be a good outcome. But what if I could edit an embryo to never have the disease in the first place? I can already do that. And in fact, in China, they're already doing that with a gene called beta thalassemia, which gives you a type of anemia. Now, there's a huge ethical debate about this, right? Some people say, well, you can't edit embryos because the embryo didn't have a choice. I don't know about you guys, I didn't choose my parents. I love you, Mom and Dad. I didn't choose my DNA. So how is this any different? I'm not really sure. But it's a big question, and it's dividing the scientific community and also the larger community. And talking about curing cancer is one thing. What if I wanted to edit an embryo to be a little bit taller, or a little bit faster, or a little bit smarter, or to have blue eyes? That's enhancements, and that starts getting into a much trickier ethical place. If we want to go even more science fiction, I'm showing you a list up here of genes and proteins that are known to make better astronauts, that let you live on less oxygen, that give you better memory recall, that make your bones stronger, that make you more resistant to radiation. We could program humans now to be the next generation of space explorers. You guys remember that movie Gattaca with Uma Thurman and Ethan Hawke? It's actually still pretty valid, except you don't sit there and go through a readout during your date. That's what the app is for. But so thinking about this, when I think about the future of humanity, space stuff is another one of my passions. This is a really interesting thought. Would I have programmed my son in order to be an astronaut before he was born? That was a question we can all be asking ourselves. And it's not just humans. We can be programming plants. We could be programming our bacteria so that whenever humans go on to the next place, they are, at least as far as we can tell, as well engineered as possible to be adapted to that environment. So this is stuff we can do now. So let's talk about the future. When I think about these big questions, I use 4 smaller questions to help me navigate when I'm watching these technologies. And so this is what I would encourage you to do as well. So the first question for any of these biotechnology-enabled solutions is there a need for it? That's actually a fairly easy question to answer in my mind. It's like a public health question, usually. The second question is, can it be done? That's a technical question. I'm a scientist. I really like technical questions. They're my happy place. The next question is it legal? That's a regulatory question. I told you about the EU ruling on CRISPR. You know, in Canada, it's actually illegal, a felony for a Canadian scientist to do gene editing. They can be put in prison for it. So our laws are not caught up to our technology. The last question is, should it be done? And that is both an ethical question and a moral question. And that is much harder to answer, because who gets to decide? If we're talking about editing mosquito populations, if you were to ask some hippie in San Francisco whether it was okay to edit mosquitoes, they would give you one answer. I'm betting if you asked a parent who had watched their child die from malaria whether you should edit mosquitoes, you'll get a really different answer. So who gets to decide? By the way, I don't have an answer for that. If you have an answer, please come find me during the break, because this is important. These questions have to be answered now. These are not questions for our children. They are questions for us, because we have the technology now to do all of these things. So I started my talk with this analogy of a cul-de-sac, and we've raised some pretty terrifying questions about all of these places that we've talked about today. And so what are you supposed to do when you walk out of here? You have all these questions now, and I bet you guys are going to go out and go looking, and you're going to find way more really interesting questions. So what do we do? And what I want to leave you with is this charge to remind you that the future isn't going to happen to you. You're going to make it. And you're going to do that by deciding to turn that cul-de-sac into a roundabout. Instead of going backwards, which, by the way, we can't do, we can't forget how CRISPR works. Instead of thinking about going backwards, we need to think about what are the futures that we want for humanity, and what do we have to do now in order to get there? And by asking questions like that, by getting your children and your friends to ask questions like that and to answer them, you're setting yourself up to be a maker of life, because life is going to be drastically different in the future than it is even today, and it's going to be affecting you more and more as time goes on. So thank you very much. It was a pleasure to meet with you all, and I'll see you at the Speaker's Corner afterwards.